The overall goal of this procedure is to record the single-unit responses in vivo from an identified population of neurons in a non-genetic model system. This is accomplished by first performing a craniotomy to expose the brain region of interest. The second step is to use tungsten wires to place the fluorescent dye in the region to which the target neurons project. Next, the dye is retrogradely transported, resulting in selective labeling of the projection neurons. The final step is to record from the labeled axon of the individual projection neuron. Ultimately, single-unit extracellular recordings are used to show how central sensory neurons in an intact circuit respond to the sensory stimulation in vivo.
This method can help to answer key questions regarding how neural circuits detect sub-millisecond differences in the timing of synaptic inputs.
The main advantage of this technique over existing methods is the ability to target individual neurons or even axons in non-genetically accessible systems.
Weakly electric fish use electric signals for communication and navigation. They distinguish signals from different fish using sub-millisecond differences in the timing of synaptic inputs. We asked how the presumed time comparator neurons in their electrosensory circuit are able to detect such small timing differences, but this technique can also be applied to other systems, such as visual and auditory sensory pathways.
It is particularly difficult to record from these cells because of their small size, dense myelin in the surrounding tissue, and the presynaptic terminals that cover the most of the cell body.
Intracellular dye injection during a whole cell recording